The scientist I chose for my biology role models project is Lisa Fortier. She's a DVM, has her PhD and ACVS, and she works in the Department of Clinical Sciences at Cornell University College of Veterinary Sciences. Her education journey is here. So she started in 1984 and attended Morehead State University as a pre-vet. And in 1989, she earned her Bachelor's of Science at Colorado State. She also earned her DVM from Colorado State. And then she completed an internship in large animal surgery and medicine at Illinois Equine Hospital. From there, she completed a residency in large animal surgery at Cornell University. She also earned her PhD and completed her postdoctoral fellowship in pharmacology at Cornell University. An interesting hobby that she has is that she competes in hunter jumpers. So she shows horses, which kind of aligns with her work too. What I admire about her is that she's a well respected woman in STEM. She's an expert in regenerative medicine and veterinary sciences, which I think is very cool. She works primarily in the field of equine medicine. That's what I want to go into. And she works with high end equine athletes. The research that she does is focused primarily on regenerative medicine, arthritis, and tendonitis. The current research that she has going on in her lab right now is mitoprotective therapy for treatment of ankle PTOA, performance evaluation of a non degradable synthetic device for chondral and osteochondral defects of the knee, mitochondrial dysfunction as a link between cartilage injury and osteoarthritis, hemiplug enhanced osteochondral repair in an ovine model, and heparin coated BMA components. The data figure I chose is right here. It's a image of mitochondria in this experiment. So the question that it addressed was, how do the mitochondria in chondrocytes respond immediately after a mechanical injury, and how does the drug SS31 affect this? The techniques used were electron microscopy, or TEM. So they took samples of cartilage and injured them by impact, and in some cases treated them with SS31 to see what the differences were. These injured cells, specifically the mitochondria structure, were then immediately studied via electron microscopy to determine the effects that the impacts had on them. The controls in the experiment were the samples, which were young bovine cartilage, and tests were done at 21% oxygen. The conclusion that came out of this was that immediately after injury, the mitochondria in untreated cells lost internal structure, so they had a poor prognosis. But in those treated with SS31, the mitochondria were unharmed. So overall, the drug SS31 helps with long-term healing, and after injury is effective for cartilage issues. Some findings and things she's discovered throughout her career. Regenerative medicine for cartilage repair, she's worked a lot with. She's performed many studies involving the use of platelet-rich plasma, stem cells, and bone marrow concentrate to improve healing of cartilage. She found that adding micronized allograft cartilage, or biocartilage, in addition to platelet-rich plasma to microfracture procedures in horses produces better repair. For cellular mechanisms, she did work on aging and deceased chondrocytes. She found that aging and disease changes chondrocyte response to growth factors, which has a connection to proteins. This provides an explanation for why cartilage has a harder time healing in older joints. And for next steps, she's continuing her research primarily regarding regenerative medicine in equine athletes to help with injury repair. Thank you for watching.